Well, welcome everyone once again to the AMS monthly seminar. I'm Jonathan Plett. I am the president of the society. And it is my pleasure this month to welcome to speak to us Felipe Albornoz, who's going to be speaking about his work in a vast area or in a very complex area that we really need to delve into more. So Felipe is interested in that interrelationship of climate and soil, plants, and then as well as microorganisms and how they fit in there. He did his PhD here in Australia at the University of Western Australia, and then did postdocs in Oregon State, and then came back to Western Australia to do another postdoc, and is currently at CSIRO. So he's published in a wide variety of areas. He's published into how pathogens impact on plant competition. He's looked into that really cool chronosequence in Western Australia, looking at how different beneficials, ectos, as well as arbuscular mycorrhiza are impacted across that whole cross-section. And today he's going to be talking about how ecosystem degradation now is going to be thought or is impacting some of these beneficials, and specifically looking at arbuscular mycorrhizal fungi. So with that, I will turn it over to you, Felipe.
Thank you. Thank you everyone who's listening. I first would like to begin by acknowledging and pay my respects to the traditional custodians of the land where this research was completed and from where I'm presenting today, which are the Wayuk and the Yaqui Yaqui, and the continuation of their cultural, spiritual, and educational practices. I first like to begin with a fun fact. Did you know that there is a connection between cow poop, mushrooms, and aliens? Anyways, fun fact. Today, I'm going to be presenting to you how different types of ecosystem degradation can have very contrasting effects on arbuscular mycorrhizal fungi. The main focus of this research is on this group of fungi that are arguably one of the most important plant symbionts. They're called arbuscular mycorrhizal fungi, or AMF for short. They're characterized by the formation of arbuscules, these tree-like structures which are forming the root cells, where nutrient for carbon exchange happens. In these arbuscules is where the plant gives the fungus carbon from photosynthesis in exchange for the fungus giving the plant nutrients, namely phosphorus or other benefits. But AMF have many symbiotic and ecological roles that are critical for ecosystem health, like nutrient acquisition, as I mentioned, namely phosphorus, which is what they're mostly well known for. But they also offer drought-resistant pathogen defense. They increase nitrogen fixation by legumes. They are involved in plant facilitation. They increase soil aggregation and carbon sequestration. And they contribute to an overall healthy soil microbiome. So you see that they are incredibly important for ecosystems, and they warrant research. To the point that a 0.05 second Google research yields almost 200,000 studies on AMF. So they're a huge line of biological research. But now to the problem, ecosystem degradation. We know that ecosystem degradation alters soil chemistry and nutrient cycles, which in turn can disrupt ecosystem processes like carbon fluxes, which can even affect climate, ultimately threatening biodiversity. But as I show you in this last slide, we know that AMF are involved in many of these processes. So what is the effect of ecosystem degradation on AMF? Well, that's a great question, and this presentation is going to focus on this. Now, what do we know about the effects of ecosystem degradation on AMF? Short answer is complicated. There is great discrepancies in the literature regarding these topics. Some studies show that ecosystem degradation can increase, decrease, or have no effect on AMF abundance or diversity. But what could be the sources of these discrepancies? After some extensive literature research, we narrowed it down to at least two potential things. First are the characteristics of the original ecosystem prior degradation, meaning what were the dominant plant groups? Was the ecosystem naturally nutrient poor or naturally nutrient rich, etc.? Let's take, for example, the lowland heathlands in the Northern Hemisphere. These systems can be dominated by plants from the Ericaceae family. And as you can see from these pictures, the plants with the pink flowers can almost completely dominate the landscape. But these plants do not associate with AMF. They are associated with another group of mycorrhizal fungi. So in this natural ecosystem, excuse me, there wouldn't that many AMF, right? Because there just aren't that many potential hosts. But degradation of these ecosystems, driven by nitrogen deposition, is allowing the invasion of AMF-dependent grasses. So all of these green grasses that you see in this right picture, they're exotic invasive plants that rely heavily on AMF. So in this case, ecosystem degradation could potentially promote AMF, right? Because we're increasing the abundance and diversity of potential hosts. But now, what would happen if we have the complete opposite? A typically AMF plant-dominated natural ecosystem that is invaded by non-mycorrhizal plants. This will likely have the opposite effect on AMF. This is above ground. Below ground is a little bit more complex, which brings me to the second potential reason for discrepancies in the literature, which is the nature of degradation. Meaning, are we talking about nutrient enrichment, like nitrogen deposition? Are we talking about the opposite, nutrient depletion, or plant invasions, different types of land use, fires, etc.? For example, going back to the heathland example, nitrogen deposition is what was allowing the invasion of AMF plants. But what would happen if degradation is nitrogen depletion? Unlikely that the same exotic plant species would invade. Now, let's put these two ideas together, the characteristics of the original ecosystem and the type of degradation, into a theoretical framework. We're using Nancy Johnson's theoretical framework from 2010, where she proposes that effectively it is the nutrient limitations between nitrogen and phosphorus for the plants that are ultimately going to drive AMF. What does this mean? For example, if we have an ecosystem that is naturally phosphorus and nitrogen poor, plants are going to be allocating substantial amount of carbon to below ground acquisition of both nitrogen and phosphorus. But if we have degradation, once again, in the form of nitrogen deposition, this nitrogen enrichment is going to exacerbate the phosphorus limitation simply by the law of the minimum. The more that you have of everything else, the more limiting becomes whatever you have the least of. And this is going to trigger the plant to allocate more carbon to phosphorus acquisition, meaning more carbon to AMF, potentially having a positive effect on AMF. Now, on the other hand, if we were to have an ecosystem that is naturally phosphorus rich, plants will be allocating mostly carbon to nitrogen acquisition. But then if we have the exact same ecosystem degradation, so nitrogen deposition, this nitrogen enrichment is going to end up reducing the overall nutrient limitations for the plants because it was already getting all the phosphorus that it needed from the soil without needing to pay too much carbon to AMF. And now it's getting all the nitrogen that it needs. So this is going to result in less carbon to below ground nutrient acquisition, which means less carbon to AMF, potentially being detrimental to AMF. And we would expect the opposite, or at least something different, if instead of nitrogen deposition, we will be talking about nitrogen depletion. So this is what I mean that in theory, both the characteristic of the ecosystem, phosphorus rich or phosphorus poor, and the type of degradation, nitrogen enrichment or nitrogen depletion, may have a hand in how AMF responds to degradation. And this is what we aim to test. So what do we do? We conducted 2 independent studies in temperate eucalypt woodlands, one in the box gum woodlands in the east coast of Australia, and one on the York gum woodlands in the western part of Australia. Now, even though these 2 ecosystems, these woodlands are eucalypt woodlands, they're quite different. They are different climate, different plant species, different eucalyptus species, slightly different soil chemistry, etc. But the one thing that they do have in common is that they're typically phosphorus limited, as most ecosystems in Australia are. And in each of these independent studies, we evaluated soil chemistry, vegetation, and AMF communities through DNA sequencing across different types of degradation. First, I'm going to show you the results of the box and woodland, so in the east part of Australia, and then I'll show you the results of the box and woodland in the west part of Australia. Now, what do I mean by different types of ecosystem degradation? Prover et al. In 2002 developed a state and transition model to characterize vegetation or ground layer states, depending on the type and severity of degradation. But because of a time constraint, I'm not going to get into too much detail. I just wanted to show it to you to let you know that we use 4 of these degradation states for experimental design. We first have our reference data, what would be our control, which are pristine box and woodlands with little to no degradation, little to no exotic plants. These are typically low in nutrients. They have very high native cover and very little, if any, exotic plant cover. Then we have a nutrient depleted state where carbon and nitrogen are significantly lower in comparison to the reference, and they have a little bit more exotic plant cover. Then we have the complete opposite, a nutrient-enriched state where nitrogen and carbon were heavily enriched and exotic plant cover is very high and native cover is very low. And finally, just for good measure, we added an intermediate state between the reference and the enriched, where nutrients and exotic plant cover are elevated, but not as much as the enriched state. And what do we find? We're excited. This plot shows each ground layer or degradation state on the x-axis and AMF richness on the y-axis in the form of 0 operational taxonomic units or COTUs. Something I want to note is that we have 2 depleted states because some of them were dominated by one particular grass and others were dominated by another grass species. So just in case this has some sort of strong effect on AMF, we decided to keep them separate. The first thing we found was that AMF richness was higher in degraded states than in the reference states, but it was by far the highest in the intermediate and the nitrogen-enriched state. Now, what does this mean? Does this increase in richness with degradation means that are we finding more number of the same AMF species per sample, or that new AMF species are arriving with degradation? We found that there is actually both loss and introduction of AMF with degradation, meaning several OTUs were lost in ecosystem degradation, meaning they were only in the reference state, but also several OTUs were unique to degraded states and not found in any of the reference sites, hinting maybe this new AMF that may not be native. So we're calling them weedy AMF for now. I would like to note also that this gradual increase with AMF richness across degradation state was also associated with changes in community composition. Not shown here for time, but I can show it to you at the end. Yeah, so to understand what might be driving this increase in richness and changes in community composition with degradation, we conducted a structural equation analysis where each degradation state was transformed into a binary variable, so 0 or 1, and the reference state is implied when all degradation states are 0. This is a very complex figure, so I'm just going to walk you through the most interesting result. That we found that the enriched and intermediate states, the one that had the most AMF richness, indirectly promoted AMF richness by first increasing soil fertility, which in turn promoted exotic cover over native cover, represented here by the native to exotic cover ratio. And this, and only this, had a direct effect on AMF richness and changes in community composition. This is very interesting because if we would have only looked at soil chemistry and forget about plants, we would have thought that soil fertility was having a direct effect on AMF, but this is not the case. The increase in soil fertility prompted by degradation is promoting plant invasions, and these plant invasions is what is promoting AMF richness. So what does this tell us about the native bogs and woodlands? Probably that they're naturally poor in AMF diversity. These woodlands are phosphorus limited, so they have likely developed more efficient nutrient acquisition strategies other than AMF, while exotic plant species are well known to be very AMF dependent, at least the exotic plants invading this ecosystem. So what are the take-home messages that from this study? First is that we support Johnson's hypothesis that nitrogen enrichment of phosphorus-poor soils promotes AMF, likely due to an exacerbation of phosphorus limitation, because we are adding nitrogen, making phosphorus more limited. But it's not that simple, because we've also found that a direct link between exotic plant covers and AMF richness, which means that it is likely that the increased phosphorus limitation of the arriving exotic plants is what might be driving this, which is very surprising. You learn something new every day. But now, what are the implications for this? So new questions arise from this study. First, there are the implications for biodiversity. Does ecosystem degradation always hinders diversity? Because in this case, kind of promoted AMF diversity. But so what do we do in those cases? Or do we care about that? Do we care about microbial diversity, or we only care about plants and animals? The second question is, where do these new AMF come from? Are they native? Are they exotic? Etc. And lastly, yes, we support a part of Johnson's hypothesis, but would we support it also when both nitrogen and phosphorus are enriched? My screen froze. There we go. She's going to mention that this study has recently been published in Plant and Soil. And if anyone would like to see it, there it is for 1 second. Now, these questions take us to the other side of the continent, to the Yorkham woodlands in Western Australia. In this study, the design was very similar, but now with the knowledge of the bogs and woodland study. So we wanted to dig a little bit deeper. We wanted to create a one-directional gradient of degradation based on exotic plants and soil fertility. Something to note of the Yorkham woodlands is that they're very, very phosphorus limited. And in some cases, phosphorus and nitrogen co-limited. Once again, we had our controls, which are our reference sites in nature reserves that are quite pristine, with little to no exotics or any soil alterations. Then we had our weakly degraded state, where exotic diversity is slightly higher, but soil nutrients are relatively similar to the reference state. Then we had our moderately degraded state, which are nitrogen enriched and heavily invaded by exotic plant species, similar to the nitrogen enriched state from the previous study. And finally, the novelty of this study, to go beyond the bogs and woodland, is the highly degraded state, where both nitrogen, phosphorus, and other nutrients were highly enriched, and exotic plant diversity was very, very high. Now, to do this experimental design, we made use of a long-term field experiment that is part of a global consortium called the Nutrient Network, or NatNet for short, where they have been adding different nutrients in different combinations for many, many years. To our fortune, this experiment is located right next to a fantastic Yorkham woodland nature reserve called Mount Caroline Nature Reserve. Sadly, this reserve has some patches where nitrogen is enriched and have been heavily invaded by weeds, but these patches seems to be very self-contained. So we made use of both of these assets to create our experimental design. So once again, our reference were the pristine areas of the nature reserve and at least 20 meters away from any invaded patch. Then our moderately degraded state, the nitrogen-enriched state, were these invaded patches inside the nature reserve, where we know nitrogen is highly elevated and exotic diversity is high. Then our weakly degraded were the controls of the nutrient addition experiment, so where no nutrient has been added throughout the experiment. But because it's outside the nature reserve, it's a bit degraded, so exotic diversity is a bit higher than the reference. And then the heavily degraded state was the fully fertilized treatment of the nutrient addition experiment, where phosphorus, nitrogen, micronutrients have been added persistently every year. So we create this nice gradient in respect of increasing nitrogen, phosphorus, as well as exotic cover and diversity. And what do we find? Very exciting results. Very, very similar results to the previous study. This graph shows each degradation state on the x-axis in an AMF richness on the y-axis. And once again, we found that AMF richness was highest in the moderately degraded state, the one that is invaded and nitrogen enriched. But interestingly, the degradation state with by far the lowest AMF richness was the highly degraded state, the one that is nitrogen, phosphorus, and heavily invaded. And it was almost 3 times lower than the reference state. Now, to go a bit further, because in the box and woodland, we had a hint that there was a different response between the native plants and exotic plants, we wanted to actually test this as well. So we assessed the abundance of AMF, measured as percentage of root colonized on the left panel, as well as richness of AMF, the right panel, between native and exotic plants across all sites. And you can see that both abundance and richness was significantly higher in exotic plants than in native plants, confirming our suspicions from the previous study that native eucalypt woodlands in Australia rely on AMF far less than exotic plants, potentially explaining why are we finding more AMF in degraded sites. Degraded sites have more weeds, hence more AMF. However, it's not that simple again, because the highly degraded state is almost completely dominated by exotic plants. Exotic plant diversity is the highest here, yet it showed the lowest AMF diversity. So exotic plant diversity alone does not explain AMF richness. It has to be this combination of both nutrient enrichment and exotic diversity. Now, I want to show you the raw data, actually. These are two-way tables that show the raw data, and I wanted to show it to you because they're a great way of visualizing what is driving changes in community composition. So here, each panel shows the same thing, but separated by taxonomic family of AMF. The x-axis shows each replicate sample of each of the degradation states, and each tick on the y-axis is one AMF and COTU. And the size of the bubbles is proportional to the relative abundance. The first panel, the top left, shows all AMF together, and then in the subsequent panels, I split them by taxonomic family. And I want to show you the interesting thing that the moderately degraded state, so the nitrogen-enriched state, invaded, has a large number of unique OTUs that only occur in this degradation state, suggesting that they are indeed aliens or exotic AMF, because otherwise we would have found them in the reference state as well, at the very least in very low relative abundance. Because we're talking about just a 20-meter distance between a reference and a moderately degraded state, so it's not that far. And now for the taxonomy fans, I just wanted to show you that these unique OTUs that only occur in this degradation state belong to the Acaulosporaceae family, to the Diversisporaceae family, the Glomeraceae family, and less so to the Pasisporaceae family. Now, this disappearing and appearing of particular groups of AMF among degradation states raises the questions about their functional diversity. Are these weedy AMF functionally redundant? Are they increasing the functional diversity of these plots? Are they outcompeting native AMF? or they're not doing much? Who knows? Those are great questions. Yeah, so again, wanted to dig a bit deeper to check what is this relationship between degradation, soil fertility, plant and plant invasions in influencing AMF, similar to what we did in the box and woodlands. These are the results of a structural equation model, but now this time, because we had a one-directional gradient, degradation states were transformed to an ordinal variable from 0 to 3, 0 being the reference, 3 the highly degraded state. Again, these figures are quite complex, so I'm just gonna show you the cliff notes. We found that, again, degradation was indirectly promoting AMF richness and changes in community composition by first promoting soil fertility, which in turn promotes exotic plant diversity, and it was exotic plant diversity that had a direct positive effect on AMF richness and community composition. The only exception was the highly degraded state, the nitrogen, phosphorus, and invaded state, that phosphorus was so high that it has its own negative effects on AMF richness. So what does this tell us about the York woodlands? Once again, that they're probably naturally poor in AMF diversity, likely because native vegetation, because I showed you that vegetation doesn't associate with AMF and is as much as exotics. So it's the arrival of weeds prompted by degradation that is promoting AMF, but this is constrained by the type of nutrient enrichment. Nutrient enrichment allows the arrival of AMF-dependent exotic plants that then they find themselves phosphorus limited, promoting AMF. But both nitrogen and phosphorus enrichment also allows the arrival of exotic plant species, but these have no use for AMF, decreasing AMF. So what are the take-home messages of this second study? First, once again, that we support Johnson's hypothesis, but now we can support the second part, that when you have both nitrogen and phosphorus enrichment, this is going to hinder AMF. Then we confirm that exotic plants associate with AMF far more than native plants. Hence, degradation can promote AMF diversity via plant invasions. And lastly, it seems very likely that this increasing AMF richness is due to the arrival of potentially exotic AMF. So these more questions arrive. What are the implications for restoration then? Can we manage with the AMF? Is that even possible? Can you manage an exotic soil microbe? But if we could, since we now have a better understanding of how AMF responds to different types of degradation, we can propose some potential managements. Meaning, if you have that graded ecosystem where nitrogen is enriched, we might have to manage this with the AMF, find some way of removing them. But on the other hand, if your degraded ecosystem is both nitrogen and phosphorus enriched, we might need to reintroduce native AMF into the system because there's a clear drastic loss of AMF diversity. These are great questions for future research. I hope somebody here is excited to do it. So the final messages of this presentation, if there is anything I want you to leave knowing. The first is that we must start looking at the whole picture with studying microbial ecology. We cannot just ask what is the relationship between plants and microbes without an environmental context, in this case, the soil. We cannot just study the relationship with soil chemistry and microbe without assessing the plant communities because it is the microbe's host. All these three components are intrinsically connected and they need to be accounted for. The second thing is the characteristics of the ecosystem matters, whatever you're studying in terms of microbial ecology. If you find yourself with your results are different and you're trying to compare them with other studies, you need to account for whether your study, the ecosystem where you conducted your study is different from the studies you're trying to compare with. And on the same line of thought, the type of degradation matters. The amount of studies that I see when they talk about degradation or nutrient enrichment as a blanket statement is concerning. Because if you're talking about nutrient enrichment, and another study is developing a fire, you just cannot say that they both are degradation because they are different types of degradation. And finally, the most interesting results for us was that plant provenance matters. Again, the amount of studies that I see where there's no information of whether the plant communities are native or not, it is concerning. If you are going to study the relationship between microbial and plant communities, you have to have an understanding of the provenance of each of the plant species in your community and account for. So, fun fact, if you fertilize naturally phosphorus-poor soils, you're going to allow the invasion of alien exotic plant species, which in turn are going to alter the communities of arbuscular mycorrhizal fungi, which in turn can both further promote plant invasions and soil degradation. I'd like to finish by thank you everyone who is listening and all my collaborators and volunteers for these studies. Thank you very much.
Great. Thank you very much for that, Felipe. That was a really lovely overview. And it's really nice to see, even though you were saying that ecosystem really matters where you're studying this, to see that Nancy's hypotheses over from, North America are really being seen here in a very different environment and very different landscape. So we already have 2 questions in the Q&A. So if anyone wants to ask questions, please just pop it into the Q&A below, and then Felipe can ask or answer them. Sorry. So the first one from Adam Frew is, great talk. We loved it. We're wondering if you found any, oh dear, Adam, you're killing me, Paraglomeraceae in your second experiment. We found them in our own NutNet site that NP fertilization had distinct impacts on Paragloma species.
No, we did not find any Paraglomeraceae. However, we are conducting in another research that we're doing in the Monaro region in Eastern Australia, also different type of eucalyptus system. We did find them there. But it's not related to this, it's more related to dieback.
Cool. And then he has a follow-on question, more technical. What gene region or primers are you using when you're detecting your AMF?
Yeah, we're using the Sato et al. Primers. There's the 18S RNA region. I do not remember exactly the bloody code of each of the primers. I'm sorry.
Okay, cool. Maybe you can follow up and send him the reference if you're using, that'd be great.
Yeah.
Next question from Camille. Thanks, Felipe. Interesting talk. Love the memes. Fully agree with that one. Do you think that the increase in AMF with disturbance is linked to the loss of ectomycorrhizal diversity and abundance, since these woodlands are often dominated by ECM associations? And follow on, does plant competition drive below ground changes or competition below ground drive the plant changes?
Okay. First question was, repeat the first question.
Yep. So, and if you actually open up your Q&A at the bottom, you can also see them.
Awesome. Can I stop sharing then?
And nope, if you just go down to the bottom, you should see Q&A at the bottom of your screen, do you?
No, I only have like a little.
Oh, okay, sorry, I'll repeat the question then. Do you think AMF increase with disturbance is linked to the loss of ectomycorrhizal fungi?
It could potentially be. There is a fair bit of evidence that there is some competition between ectomycorrhizal fungi and arbuscular mycorrhizal fungi. However, the reference states and the naturally enriched states, they both occur inside the nature reserves. So between an invaded patch and a non-invaded patch, the eucalyptus cover does not change. So unlikely that is that. It is more likely that it's the arrival of the weeds.
Actually, did you look at the roots of the eucalyptus to see actually if the AMF might have been displacing the ectos, or?
No, we did not. We looked at roots of understory species only.
Okay, Right.
Yeah.
And the second question from Camille was, does plant competition drive below ground change, or does the competition below ground drive the plant composition in these cases?
It's probably both. It's probably a feedback. Yes, but what I can tell you is you do not get plant invasion if you do not get the nitrogen enrichment first. So that's why so many areas of the nature reserve are so pristine, because they are not nitrogen enriched. But as soon as you add nitrogen, the weeds arrive. And now that is indeed a positive feedback. These weeds promote the persistence of that elevated nitrogen. So they don't just suck up the nitrogen and then die. They continue that positive feedback.
Yeah.
But whether the competition between native and exotic plants drive the underground, hard to tell.
Great. Question from Anna Hopkins. So, hi, Felipe. Great presentation. Did you look at changes in ectomycorrhizal fungi as well? Do you have any thoughts about the diversity? So that's leading on a little bit from what Camille was saying as well. So did you actually quantify anything ectomycorrhizal in those?
Yeah, unfortunately, no, because of.
Yep. Fair point. Question from Tom.
I can only focus on one microbial group.
Question from Tom May. People involved in restoration, once they know about the role of AMF fungi for plant health, often ask, where can I buy these fungi? How do you answer that question?
Don't buy them. So AMF in restoration is already getting a fair bit of traction. So there are a lot of studies, and at this point, there's even meta-analysis of how reintroducing native AMF to a degraded ecosystem can promote native vegetation, so promote restoration. However, if not all of these studies are from the Northern Hemisphere, where phosphorus isn't really limiting. So here, where AMF diversity is lower, and we're seeing an increase in AMF diversity, so we're having a problem of potentially weedy AMF, why you don't really need to reintroduce anything. We need to take out, essentially, potentially.
So I was curious about the.
Sorry, it's just the second part of that.
Sorry, yep, go for
that question. Yeah. So you can buy AMF inoculum, but those are commercial inoculum based on these 1, 2, 3 species that are usually made for agriculture, etc. Every single ecosystem is going to have its own AMF communities. So I cannot go buy a restoration AMF pack from Europe and use it here. Completely different AMF communities. Even from the East Australia to Western Australia, different communities. So the way that it is done is you go to a reference site, you collect soil, and then you culture those AMF and then bring them back to a degraded site. That would be the way to do it. Morally conscious.
Yes, while avoiding the pathogens at the same time, so we don't spread them. Yeah. So you're calling them weedy AMF, and so I'm curious about that because I've used that statement myself. So why do you call them weedy? Could they not just be also beneficial? Because I mean, weedy has a connotation that you really do want to eradicate them, and I guess one of the things I was thinking is, as I was listening to your presentation is, and you had the question in there that isn't answered, are these new AMF, are they actually arriving? But I would ask the question, are they actually there at really low quantities? And then you have these new plants, and actually then they're like, oh, I can, be hosted by that plant. And then they just go crazy.
Yeah. So 2 things.
Yep.
First, the fact that we did not find a bunch of those AMF OTUs in any of the reference sites, and the reference sites have no exotics, it's very likely that these new AMF are very host specific to exotic plants. And if they're host specific to host exotic plants, it means they have to have come from where these exotic plants come from.
Yep.
But again, with our results, we cannot say definitely beyond reasonable doubt that there are exotic AMF. So that's what we call them weedy AMF, because at the very least, they benefit from degradation.
Fair. Yep.
So they thrive in degraded soils. Whether they're native or not, they thrive in there, like a weed will do.
So Tom has a follow-on from question from your answer. He loves your "don't buy it" answer. Are you making any collection of these native AMF to allow people to actually match ASVs to specimens?
Yes, that really hurt whoever asked that question. We have that planned, and that as we want to do, we were going to start in May to do so. Start collecting from everywhere, isolating, making pure cultures. But my microbial growth chamber hasn't arrived, and it's not going to arrive until November. Oh, the plan is to do so, and it should have been done so by now. But thanks for bureaucracy. I can't.
Not yet. Yes, Tom May is always after me for collections in my own fungi. So
but harass me next year. I will have them next year.
Yeah, that'd be really interesting. It'd be interesting as well to see, you know, how they're interacting with eucalyptus in those areas as well. So we have potentially a final question from Salma Sarkar, who is asking, do you have any conclusions about the introduction of new AMF into degraded soil compared to reference site? So where are the new isolates coming from?
Yeah, it is likely that they came with the weeds during the colonization era, where whenever the weeds were first introduced, it is likely that they came with them. Same situation with pine trees. Mycorrhizal, that the pine trees can only invade other areas of the world only if they come with their the specific ectomycorrhizal symbiont. So it is possible that's the case, but again, I'm not fully ruling out the fact that they might be native, just very, very rare, and I didn't pick them up.
So these weeds then, are they actually slowly encroaching in there? Or because when I think of weeds depositing in some of these areas, I would think of just seeds blowing in from long distances, but that's not the case then? The weeds are actually slowly encroaching in? No,
weeds are blowing everywhere.
Okay.
So we're talking about Avena, Hypocaris, Arctotheca, your typical everywhere weeds. But the beauty of especially Western Australia is that the soils are so poor in nutrients that these weeds have no chance. So they might be throwing seeds everywhere, but they do not survive.
Okay.
Yeah. They only survive where degradation has happened.
Yep. Great. Cool. Well, I think that ends the questions that have been coming out in. Thank you so much again, Felipe, for that great presentation, for answering all of our questions. Thank you to those who have been here and asking the questions. We will be back again later in the next month, hopefully with our next seminar. Thank you very much, Felipe, and thank you to everybody who came.
Thank you for having me. Bye-bye.
Bye.